Hello dear friends, welcome back. And now in the previous lecture we discussed about DNA, right? All those basic things regarding DNA, and we had said that we will combine these three important factors, the nitrogen bases, the sugar and the phosphate, and we will make the DNA. So now let's make the DNA, how the DNA is formed, right? Hi guys, let's make the DNA, right? So structure of DNA, let me write here, structure of DNA, right? It means that how the DNA is formed, right? So let me remind a little bit the previous lecture. First of all, I had told you that in order to make the DNA, we should have three important factors. The first one is the nitrogen base. The second one, that we should have, there should be a sugar with us, and the third important thing that we should have, that is a phosphate. We should have a PO4, right? And the sugar that I had told you will be the deoxyribose, right? Now let's combine these three important things and make the DNA, right? So first of all, our steps. Let me write here steps. So step number one, that we are making the DNA. Look friends, if you watch the lecture up to the end, right, and you don't skip, you will learn the structure and composition of DNA in this one clip and you will never forget it, right? So let's focus here. The first step which happens is that what do? We combine this nitrogenous base with the sugar, right? So the first step first, fuse nitrogenous base, nitrogen base with sugar, right? So let's do this step. Okay, we have sugar, right? You know, we have sugar, there is the oxygen. Let's draw a little bit structures also. This is our friend sugar, right? This is carbon 1, carbon 2, carbon number 3, here we have carbon number 4, and then we had here carbon number 5, right? Now what do? This is our sugar, right? Okay, we combine the nitrogen base here. Here will be our nitrogen base, and I'm making it very simple friends, not the complicated structure I'm drawing, just to simplify everything, right? So if you remember, I had told you that those nitrogen bases which are present in DNA, right? How many types? The double ring and the single ring. The double ring were adenine, adenine and guanine, right? And the single ring which were present in DNA, in case of DNA, not RNA, that was the thymine and cytosine, not also the uracil. Uracil is present in RNA. So these nitrogenous bases we are combining with the sugar, right? So if you are combining the adenine, for example, you are combining the adenine here, right? So this, if this is your adenine, right? So this is the nitrogen base, right? So nitrogen base, what did we do? We combined this nitrogen base with this sugar, right? So this is our sugar, right? So this was our step number one, right? And if you have 4 nitrogen bases, so you should have 4 sugars also here, right? So this is a sugar. We make it fast. right? And we should not forget our carbon number 5 also, right? And then if we put here another nitrogen base, right? So this, if this is our adenine is done, if this is our guanine, right? Guanine. So guanine combined with sugar, deoxyribose sugar or pentose sugar, right? And then here we combine the next nitrogen base, right? That is thymine, right? And then here we combine that our next nitrogen base, and that if is cytosine, right? Cytosine, right? So what did we do? We combined the sugar with nitrogen base, right? So all of these are nitrogen bases, and here we have sugars, right? So this molecule in which the nitrogenous base is combined with this sugar, this molecule is called the nucleoside, right? So we say fuse nitrogen base with sugar to make nucleoside, not nucleotide, right? Look, so this is our nucleoside, this is nucleoside, this is nucleoside, and this is nucleoside, right? So what is the names of these nucleosides? Let's name them. Look friends, if adenine is combined with with sugar, we call it adenosine, right? So adenine combined with sugar, we say that it is adenosine. Guanine combined with sugar, we say this is guanosine, right? And thymine combined with sugar, we say thymidine, right? And cytosine combined with sugar, we say cytidine, right? Cytidine. Right? So these 4 types of nucleosides have been formed, not nucleotides, right? We are going to make the nucleotide. So we fuse nitrogen base with sugar, we made nucleosides. So 4 types of nucleosides are formed: adenosine, guanosine, thymidine, cytidine. So this is our step number 1. Now let's move towards the step number 2. Step 2, what do we do? So these 2 are done. Now what is the turn of phosphate? So fuse phosphate with nucleosides. This is our step number 2. Fuse phosphate with nucleoside, right? To make what? To make nucleotide, right? So this is our second step. Now what do? We take this nucleoside and put a phosphate. So then we make nucleotide, and then we will combine those nucleotides and we will make DNA. That's so simple, right? So let's do that. Let's put all these here, right? So either I will complete this step here, right, in order to make our video a little bit faster, right? So let's write this step also here. We have already drawn the structure, we don't need to draw these structures there also, right? So this was our step number 1. Now step number 2 also here, right? So we put phosphates. So at carbon number 5 we put these phosphates, right? So here we put phosphate PO4, right? Here we put the next PO4, right? and here we again at carbon number 5 phosphate, and here at carbon number 5 also we put the phosphate, right? So this was our step number 2. Now look carefully. Look carefully. When sugar was combined with nitrogenous base, we called this as nucleoside, right? But if we combine phosphate with this nucleoside, what happens? Here we put the phosphate, PO4, right? So what happens? This is now nucleotide, right? So in order to make a nucleotide from nucleoside, you just put a phosphate on carbon number 5. You will get nucleotide, right? So this was the next step that we did here, right? Now what do? We make the DNA. The last step, we combine. Okay, the third step, combine nucleotides to make what? To make DNA. This is the last step. Now if you want to make the DNA, you should combine all these nucleotides. It's so simple. Look here, friends. I'll draw simple structures, right? This is the sugar, right? Then here is your friend adenine, right? And I will put A, right? And here is your carbon number 5, okay? And here is your phosphate, okay? So this is our adenosine monophosphate, right? What do we say? This was our adenosine, right? And we put phosphate. So there is one phosphate, so that's why we said this is monophosphate. So adenosine monophosphate, AMP, we just call it AMP. Now if we bring this the guanosine and we will make a bond between them, what do? Look carefully. When guanosine wants to come and makes bond with this, how will guanosine come? This is, for example, this is your guanosine. Here you have G, guanosine, right? And this is carbon number 5. So guanosine will come not with one phosphate, guanosine will come with 3 phosphates, right? That's why we call it GTP. GTP, guanosine triphosphate, guanosine triphosphate, right? Now when this guanosine comes here and stays here and makes a bond here, right? So what happens? Look, here is an enzyme which will make the bond between guanosine and adenosine monophosphate. What this enzyme is sleeping. Look carefully, friends. Look carefully. This enzyme is sleeping. Now this enzyme is going to make the bond, right? What do? When the guanosine comes, guanosine triphosphate comes, it has brought 2 extra phosphates with itself, right? What happens? When it comes near, these 2 bonds will break, right? These 2 unstable bonds will break. Now when these bonds break, energy is released. Once again, look. Guanosine triphosphate has came and he wants to make a bond here, right? Now what will he do? He will lose these 2 pyrophosphates. These 2 phosphates will be lost. Now there will be, when these phosphates are lost, energy is released to the system, right? When the energy is released, this energy is utilized by this enzyme, and this enzyme is activated. Now this enzyme, by the utilization of that energy, this enzyme will fastly make the bond between these 2, right? So this will become like that, Look, here is our carbon number 3, right? This is carbon number 1, carbon 2, carbon 3, 4, and that is 5. Now you know that this is the sugar which was brought by guanosine. Now here is our guanine, right? And it had lost its one phosphate, you know, bitter, right? It has lost its 2 phosphates, right? So when the 2 phosphates gone, only 1 phosphate is remaining, right? And those phosphates which were gone, they released energy, and that energy was utilized by this enzyme, right? And this enzyme fastly made the bond between the carbon number 3 of that upper nucleotide and carbon number 5 of this lower nucleotide, right? So here is a bond established, right, by this enzyme, right? Who activated this enzyme? This enzyme was activated by the phosphate bonds, right, but the energy which was brought by this friend, right? So bond is established by this enzyme. This bond, now look, 2 nucleotides has been fused together, right? Now let's fuse another nucleotide here, right? So adenine and guanine has been fused. Let's bring this thymine here, right? When thymine comes, right, and this thymine has again brought 3 phosphates with itself, right? So we said thymine triphosphate. So this is phosphate, another phosphate, another phosphate, like this, right? When it comes near, it again breaks these bonds. So the pyrophosphates are gone, right? And the energy released. The energy is again, when this enzyme has established this bond, it will come down, right? When it comes down, it will use this energy and will make a bond here, right? Now look, what are we doing? We are fusing the nucleotides with each other, right? We had said that in order to make the DNA, we should have nucleotide and we should have fused them together, right? So this was a nucleotide, this is another nucleotide, and this is another nucleotide, and millions of these nucleotides will be fused together. Now look, this was thymine. Let's bring the cytosine, right? So when the cytosine comes, right, this is our cytosine, right? When cytosine comes, again it brings phosphates with itself. How many phosphates? It brings 3 phosphates, right? These 2 bonds are unstable, so these bonds are broken. When these bonds are broken, the phosphates are released. These phosphates go and bring more nucleotides. Phosphates go and bring more nucleotides with their self, right? So when this enzyme makes this bond, it comes down and utilizes the energy which is released here and starts establishing another bond between these 2, right? What are we doing? We are combining these nucleotides with each other, right? So and so forth, all these bonds will be made. And what are these bonds called? This red bond, which is made between 2 nucleotides, it is a very strong bond. This bond is called the phosphodiester bond, and it is a very, very strong bond, right? So the phosphodiester bond is formed by which enzyme? This enzyme is making DNA, so that's why we call this enzyme as DNA polymerase, right? So the name of this enzyme is DNA polymerase, right? So what has DNA polymerase done? DNA polymerase is making bond in between the 2 nucleotides by the utilization of energy which is brought by these nucleotides. It's so simple, right? So this is called, now if we put many and millions of these nucleotides together like this, they will make single strand of a DNA, right? Now we are going to, as you know that our DNA is double stranded like this, right? So we have made this single strand of this DNA, and now we are going to make the next strand of this DNA, right? So let's do that. Look carefully, friends. One thing I will tell you, look, here is carbon number 1, here is 2, here is 3, 4, and this is 5, right? So carbon number 3 of this strand is remained unbonded, right? And here at the top, carbon number 5. So we say this strand of the DNA is 5 prime and 3 prime, right? What say? We say that this DNA has become like this. So 5 prime and 3 prime. Why? Because here the third carbon has nothing, and here the fifth carbon has nothing with it, right? So 5 prime and 3 prime. Now what will we do here to make the other strand, right? The other strand of this DNA will be opposite, right? Why? Look carefully. In order to make the other strand, the enzyme will read all these nucleotides and will combine them upside down, right? If these nucleotides are, look, I'm drawing a very simple diagram. If these nucleotides are all like this, right? But this side nucleotides will be like this. Right? So upside down, this is the head above, and here is the head downward, right? So they will be like this, right? So upside down. Okay, let's go for it. So here let us make these nucleotides also. Let me start from here. Right? This is the sugar. Now look, this sugar and this is the difference. Look the difference. So here is oxygen, and the oxygen of it is downward, right? And now look, here is carbon number 1, here is carbon number 2, carbon number 3, carbon 4, and here is carbon 5, right? So it will be, all the sugars will be like that, right? So let me make all these sugars fastly like this, and here again the sugar will be somewhat like this, and here the sugar will also be like that. You see this? Right. So let's put the phosphates and everything here, right? This is the carbon number 5, and here is the carbon number 1, 2, 3, right, 4, and this is 5. So here is the 3 prime, and here is the 5 prime. Let's go for it. Look, you know that adenine combined with thymine, right? Adenine binds with thymine and cytosine binds with guanine. These are the complementary base pairs, right? So if you have here adenine, here you will have the thymine, right? So here will be our thymine. I'm sorry, I should make it a little bit above, like this. So here should be your thymine, right? This will be thymine, and this sugar also a little. If you have guanine here, here you should have cytosine. Here is cytosine, right? And again this will be our sugar like this, right? If you have thymine here, here you have adenine, right? This is your adenine, right? And here this is the sugar, right? Okay, if you have cytosine here, here you will have guanine. This is the G, right? And this is carbon number 1, 2, 3, 4, and this is 5, right? 1, 2, 3, 4, and here is 5. So here's 3, here is 5, here's 3, 3, and this is 5, right? And the enzymes, again, we will put all those phosphates here too, right? Right, these are the phosphates, right? And these are the bonds, the phosphodiester bonds like this, right? So now this is the one strand and this is the next strand, right? Now these strands should not be elastic, right? They should have bond. In between the nitrogenous bases we have the which bonds? We have the hydrogen bonds. So here we have double hydrogen bonds in between the adenine and thymine, but in between the guanine and cytosine we have a little bit stronger bond. So we have triple bond here, right? So we have again here 3 hydrogen bonds, and here we have only 2 hydrogen bonds, right? So now this strand cannot move in that way and this strand cannot move in that way because all of them have been coiled with each other, right? So this is one strand, this is the other strand, and this is whole DNA, and in this manner millions of the molecules of the nucleotides will combine and they will make the DNA, right? And you know that this DNA is not like this. The DNA is a coiled structure like this, right? Like this, right? So DNA is wrapped around both of the strands is wrapped around each other, right? Okay friends, so this was about the structure of the DNA. I hope you like the lecture. In the next lecture we will discuss about the forms of the DNA which are present inside the human cell, right? So see you in the next lecture. I hope you like the lecture. Till then, Allah Hafiz.